Hey guys, this is the third video for today. This video is talking about antibiotic resistance. We're also going to be talking about, and we're also going to be talking about, antibiotic resistance. We're going to talk about how antibiotic works, but at the same time, we're also going to be talking about the effect of HIV on the immune system and how it transmits. So let's get started. Now, antibiotics. Antibiotics, keep in mind, it only works in bacteria, prokaryotes, and not eukaryotes. And because of that, it's actually not. So, because of that, it's only focused on the mechanism, the metabolic pathway that prokaryotes use, not the ones that eukaryote use. Because if you think about it, and if you remember your prokaryote cells, the ribosome is 70S versus our ribosome is 80S. There's a lot of things which are quite different. Bacteria has a shell. Some bacteria have a shell. We don't. And there's a lot of things. So in this case, when antibiotics stops at, these are some examples. So inhibiting certain enzymes, so sulfa drugs will be an example of that. Penicillin will be inhibiting the ability to mix cell walls and essentially really just punch holes in their cell walls. Inhibiting protein synthesis, that's tetracycline. You might have heard of that. So in this case, that's how antibiotic works. It doesn't work on viruses simply because viruses don't use those mechanisms. In fact, viruses, they inject their genetic material. Now here it says DNA, but it really is its genetic material, DNA or RNA, into the living cells and use the cell mechanism, so host cell mechanism. So it doesn't really work. Antibiotic doesn't work that way. Antibiotics might be prescribed because sometimes a virus infection might cause secondary bacterial infection, such as you catch a flu and then next thing you know, you got pneumonia, that kind of stuff. So in this case, or make you more susceptible to bacterial pneumonia. So in this case, what am I talking about? Antibiotic doesn't work on viruses. There are antiviral drugs that can target viral enzymes. So HIV antiretroviral drugs, we'll see that a little bit later on. But just to looking it up right now. So right now, coronavirus, they're trying to develop an antiviral medication for it. This, as some experts say, this might be a faster way than a. This might be faster being developed than a vaccine, but we don't know. If someone can tell me, then it will be awesome because I'll be investing into stocks. But anyways, yeah, coronavirus. They're trying to develop an antiviral drug. Now, Tamiflu. I don't know any of you guys might remember it. Tamiflu, that's a name for it, is a drug to antiviral to treat influenza. So if you remember SARS or H1N1 virus, the outbreak happened. People were talking about getting Tamiflu. So that's what it is. Some of you might have heard of it. So those are examples of antivirals, okay? Regardless, antivirals or antibiotics, you're talking about not attacking the um, host cell pathways, okay? Now I'm gonna move myself over here and shrink myself a little bit because we're going to talk about antibiotic resistance. And keep in mind, this is really going back to first unit when we're talking about, because first unit we talk about natural selection and we talk about evolution. So if you remember, antibiotic resistance was a case example of that, which I did ask you to previously memorized. So I will kind of go a little bit faster on this part. But here, this part here that talks about why we have antibiotic resistance. I think someone told me about this. I think you guys learned about some of this back in ICE in grade 9. So, overprescribed antibiotics, especially the broad spectrum drugs, are being misused. Um, Now, by the way, broad spectrum antibiotics. Antibiotics generally have two kinds. Well, there are many kinds, but when we're talking about broad spectrum versus narrow spectrum, so broad spectrum antibiotics are generally the type which um, are generally the type that will kill a large group, large, yeah, large group of bacteria. They don't really care what type it is, but they will kill a whole bunch of them. Versus the so-called narrow type, narrow spectrum antibiotics, which will be focusing on a few different variation of bacteria. So you're talking about basically kills every microbe versus a few smaller type. So as you can imagine, if people keep using the broad spectrum type, which in some way is a very lazy way to treat, it could be lazy way to prescribe antibiotics, but at the same time, that also makes it easy to become resistant to. So antibiotics are freely available without prescriptions or are included in livestock feed. That's a big, big problem. Multi-drug resistant are especially common in the hospital where antibiotic use is high. Yes, and I think I told you about this. A friend of mine a few years ago caught MERS, a multi-resistant Staphylococcus aureus, in Hong Kong a few years ago. So now, this again, you have seen this back in unit one. So again, I'm going to talk about broad stroke. You should remember this, or this should be a review. So you need to first have, you need to have bacteria that have certain variations, okay? You need to have variation first. So some bacteria will carry the gene to be resistant to an antibiotics, okay? Might be mutation, might be sexual reproduction. But if there's no application of antibiotic, these bacteria have no advantage, and may in fact be at a disadvantage. The reason why it will be at a disadvantage is why on earth produce a certain why on earth to produce a certain protein which takes energy and resources if it's not really useful or necessary. Okay. So in this case, environmental change will be the application of an antibiotic. So it will kill off the ones that are susceptible to the antibiotic. The resistant one will survive and strive. So results of natural selection, resistant the population will grow. So you learned this back in unit one. Again, I'm not going to spend more time on it. Before I move on, pause. Are there questions? No? Okay, so we're going to move on. Now, this here is a history lesson, so to speak, of how penicillin, the first antibiotic that was discovered, what were the experiment being used to test antibiotic on tested by antibiotic on mice. So if you look at it, this is the fungus that produced antibiotic. This is the area of no growth, also known as zone of inhibition. And they were actually just trying to grow bacteria. Now, I don't know if you guys are going to be able to do IA this year, but usually growing bacteria as an experiment, you always run the risk of having fungal contamination, mold contamination. It is very common. Um, so in this case, just read this section. They have asked questions on this before. Make sure you know it, not just multiple choice. Sometimes there'll be short answer questions. So just read this part. It's not hard. Okay. Um, Now, the effect of HIV on the immune system and method of transmission. Um, Now, as I mentioned in my first video or the Q&A video, HIV attacks the helper T cells. So some people are asking like, hey, how important is the helper T cell? The helper T cell that we talked about here. Okay. How important is this helper T cell? The helper T cell is actually very important. Helper T cell um, activates activates the B cell. Okay, so this part is actually for the activation purpose. So without it, um, it won't work. Now, some of you are wondering, like, hey, what was this whole thing about? So, um, now this is, if you want to look like this is a study guide, chapter 11, which is the higher level stuff. I'm going to just use this. Standard level students, don't worry, but it just helps you to understand this process more. So the activated helper T cell binds to a B cell, so that's inactive, binds to it, and send the signal to activate the B cell. Yes, I know there. I talked about other stuff before, and that diagram that we showed earlier on the handout, it seems like, hey, this looked like it can be activated through a different pathway. For now in this course, keep in mind, I keep on saying this, immunology is easily a it's easily a third year course. So right now we're just talking about the immunology that high school students can understand. So this is a simplified version. Okay. So for now, so for now, um, helper T cell is, yeah, HIV only attacks helper T cells, so helper T cell is important to activate the B cells. Okay, so HIV is, this is what it stands for, human immunodeficiency virus. So usually, if you show that if that person is HIV positive, means they have the antibody in their blood. Okay, it means that it doesn't mean that the person is cured. It just means the person have HIV antibody in the blood. But the problem is that these individuals are usually infected. I know I talked about usually if you have immune antibody in the blood, usually you'll be okay. But in this case, because it is attacking, HIV is attacking the helper T cell. And also another thing, which is not part of the notes, HIV virus mutates very quickly. So while your body might produce antibody for one type of HIV or one strain, one substrain, so to speak, next thing you know, it might mutate and might not work. It is really actually draining, horribly draining on the body resource system, HIV virus. It's scary. So, but for this, for high school right now, what you do need to know, attacks a helper T cell, it binds to the receptors on their surface so that it actually can enter into the CD4 cell. So you might I think there might be one or two cases of like people who are cured or whatnot, or there are certain people who are um, resistant or immune to HIV virus simply because in some reason their helper T cells does not have certain receptors, and because of that, there's no way for the HIV virus to get in. But I'm slightly off topic, but look it up. So now retrovirus. Now retrovirus is a type of virus which they use RNA for their genetic material. Currently, the coronavirus is also a retrovirus, so they also use RNA. So once they go in as a RNA, they'll need to go for a process of reverse transcription. You'll learn about transcription probably in grade 12. Transcription is when you have DNA going into RNA. That's transcription. When you go reverse transcription, you go from RNA back to DNA. So as you can see, you have RNA go to DNA. That's reverse transcription. You'll need a particular enzyme called reverse transcriptase. Okay. Now these things are incorporated into the host genome. Cell that can make more copies of the virus, keeps doing that. As a result, the immune system is weakened. The number of helper T cells starts to decrease, as well as antibody production. Once CD cell count drop below critical value, the individual has AIDS and becomes susceptible to opportunistic infections, which can be lethal. So basically, something very simple, such as a cold or flu, could kill you. Also, another thing, opportunistic infection. Our body currently, my body, your body, and everyone's body, have tons of bacteria and viruses covering it in and out, inside and outside. The reason why we're not sick is because our immune system is working. So our macrophages might eat it, or we might have memory B cells and stuff like that. But when you're immunocompromised, that's when these bacteria and virus and even fungi could just crop up and show and cause infection. One of the ones that is common, I remember my professor talked about it, was the Candida albicans yeast. So our body usually have a lot of these yeasts called Candida albicans yeast. And that's the same kind of yeast for ladies who got yeast infection. That's the same one. So apparently a lot of that could cause an infection on the back of the mouth because of these yeast. Basically, you have yeast infection in the back of your mouth once you have a certain level. So those are examples of opportunistic infections. Now, transmission of HIV, pretty sure you heard of it at this point. You can read this yourself. Okay. Now, that's it on the material and resources. Pause here again. Do you have any questions? Now, SL students, this is the last bit of the SL material before the test. I still have to give it some thought in terms of how the test will look like. But as it stands, it will be early next week. I do want you to try these you try questions and whatnot you try questions and whatnot. And make sure you do some practice. I will also think of what we're going to do next lesson, whether it's a review, whether it will be practice on these database questions. So anyways, that's it for today. If you have any questions, make sure you continue to email me. I'm really impressed by how well you all did. Now, some people who, if you haven't done the episode video yet, make sure you do them. I'm gonna be sending emails very soon, so make sure you get that done. Okay. Bye now.